or FISH, allows you to directly visualize chromosomal anomalies that are too small to be seen in a karyotype. You start by preparing either a tissue section or cells on a slide, and then add DNA probes that are complementary to the sequences that you're interested in. Most cells will probably be in interphase, so they'll look like this one on the bottom, since the chromosomes aren't condensed, but cells in metaphase, which you'll be more likely to see in rapidly dividing cells or in cell culture, will look like this example on top, which can be more informative. Since you can label the probes with different colors, you can detect multiple sequences at the same time. You can even use lots of different colored probes to do what's called chromosome painting, to make a chromosome look like a rainbow in which each color corresponds to a different region. Then, you'll put your sample under a fluorescence microscope that can detect specific wavelengths of light. If the sequence you're looking for is present, then you will see the color or wavelength of light that corresponds to the oligonucleotide probe that's complementary to it. So here we have a green probe and a red probe that are corresponding to two different regions of the genome. If the sequence is not present or is mutated in such a way that prevents the probe from binding to it, then you won't see a fluorescent signal, and you'll know that that sequence is not in your sample. Normally you expect to see 2 copies of each probe, one for each chromosome. But if one chromosome has a deletion, then you'll only see one signal. So in this example, we're using FISH to look for the 22q11 locus, and we see that this patient only has 1. So what did we just diagnose, and what symptoms would you expect this patient to have? Well, this could be DeGeorge syndrome or velocardiofacial syndrome, and this patient might have a cleft palate, abnormal facies, thymic aplasia, cardiac defects, and hypocalcemia due to parathyroid aplasia. The mnemonic you can use to remember this is Cash22, but I'll talk about this more in the genetics section. By the way, FISH is the main way you detect this, and affected patients are heterozygous, so they have 1 present copy and 1 deletion.